Hello, my name is Patrick Allard. I'm an associate professor at UCLA in the Institute for Society and Genetics. And today I'd like to give you a short presentation on a work that aims at dissecting the mechanisms of inheritance of environmental information across generations. One of the larger questions that we're asking in our lab is, what are the molecules of inheritance? Of course, DNA is one of them. We've known that for a long time now. But it's also increasingly clear that the epigenome, so molecules that are added on or around the DNA, can also be a vector of information across generations. One of those modifications is DNA methylation, so the addition of methyl groups on cytosines on the DNA itself. It's been, as I said, a large focus of investigation for many years now. But it's also increasingly recognized that other marks, such as microRNAs or other small RNAs, as well as histone modifications, where there's a large repertoire of histone modifications, can also be a vector of information across generations. And in our lab, we're going with a hypothesis that histone modifications are required to carry environmental information across generation. And this hypothesis was really born out of the fact that there's organisms like the one that I'm using, C. elegans, that does not have DNA methylation and yet sees transgenerational inheritance triggered by different types of environmental stimuli. So while the focus historically has been on DNA methylation, it was also very important to look at other types of epigenetic modifications for their role in understanding the transfer of information, environmental information across generations. So as I said, the model that we're using in the lab is C. elegans. It has many interesting features. The fact that it's transparent is a very big advantage for the question that we're asking. It has a very short generation time and a very important feature as well. It has a very large germline. It's small enough to be used in high throughput screens. And in general, there's a great deal of conservation of the pathways that dictate reproduction, genetic pathways in general, as well as epigenetic pathways more specifically. As I mentioned, it does not have DNA methylation, but instead it has methyl cytosine methylation, type of methylation. But instead, it has a very large repertoire, conserved repertoire of histone modifications, as well as small RNAs. And for us, again, that was a great advantage because we know that this crosstalk between DNA methylation and histone modifications, and here we could have a system where there's no crosstalk between DNA methylation and histone modifications because there's no 5-methyl cytosine methylation. In terms of histone modifications, we know that they're highly conserved across phyla. So here, on the left, I'm showing the primary sequence of histone H3 between C. elegans at the top and histones at the bottom, and human histones at the bottom. And not only do you see that the primary amino acid sequence is conserved, but also the epigenetic modifications that are added onto those histone tails are also conserved, which really highlights the fact that it's not just the histones that are conserved, but the entire pathways that regulates the addition, the removal, and also the reading of those marks are highly conserved across phyla. On the right, I'm showing the effects of different types of histone modifications, and for the sake of time, I'll be focusing on these 2 marks at the bottom, histone H3K9me3 and H3K27me3. So what that lingo means, K9-trimethyl, for example, means the addition of 3 methyl groups on position 9, which is a lysine of histone H3. And we know that these 2 marks, in particular K9 and K27me3, associated with chromatin silencing, meaning a higher level of compaction of the DNA and a lack of gene expression. In terms of chemical agents that we used, we focused very quickly on studies on BPA, Bisphenol A, which is widely used in the plastic industry. At high doses is a clear reproductive and developmental toxicant, and also has been shown to alter the epigenome in many different contexts, including in germ cells. But until our work was published, we did not have solid proof that BPA by itself could cause transgenerational effects. The way we do our work and our transgenerational studies are very similar to the way they've been done in mammalian systems. We expose the worms at the parental generation, the P0, to different types of environmental agents, including BPA. We do this for 48 hours, and then we wash the worms from the chemicals, and then we follow each generation all the way up. You'll see some studies were done all the way up to the F7. And the advantage of the worm, because of its fast generation time, is that instead of the many months that it takes in mammals to get to the F3, we get to that third generation in just 2 weeks. Now, it's important to look at the F3 for studies, because when we expose a worm at the P0, the F1 is represented as embryos within this P0, and then the F2 is represented as precursors of the germline within those embryos. So any health effects that you may see in the F1 or the F2 may be caused by this direct exposure. So if you want to look at this transfer of information across generations, we have to start by looking at the F3 and beyond the F3, of course. So I'm only going to give you snippets of a work that was published in Cell Reports last year. When we did this ancestral P0 exposure to BPA and then looked at the F3, where we know that by using a sensor, things are not normal at the level of the epigenome. And then at the F7, where things seem to have gone back to normal. And we look at reproduction, we've seen that the number of fertilized egg produced, so embryos, is about the same, whether you've been ancestrally exposed to BPA or to DMSO control at the F3 or at the F7. However, when we look at the quality of those embryos, that's where we see a difference. We see an increase in embryonic lethality when the worms have been ancestrally exposed to BPA compared to DMSO. And it still trends by the F7, but it's not statistically significant. When we look in the germline of the worms that give rise to these embryos here, and we look specifically at this region of the germline, and this will be important a little bit later on in the talk, we see a dramatic increase in the number of abnormal germline nuclei that die by apoptosis. So all these green bright dots here represent apoptotic nuclei stained by acridine orange. And we see this significant increase at the F3 that is not significant anymore by the F7. So that was the first correlation, a correlation between this BPA exposure and decreased reproductive function at the F3. Now, what about the epigenome? I mentioned that we use an epigenetic sensor. Again, that's in the paper. What I'm going to show you here is the more targeted assays that we did to assess the levels of different marks. We actually used an unbiased screen at first to look at many different histone marks, and then focused on 2 of them that seem to be altered the most, H3K9 and H3K27me3, the marks that I mentioned earlier. And so what I'm showing you on these panels on the left here are the levels, global levels by immunofluorescence of K9 and K27me3 in worms that were ancestrally exposed to BPA versus DMSO versus water. And if you look on the rightmost panels, you can see a dramatic decrease in K9-trimethyl and K27-trimethyl in these worms when they were ancestrally exposed to BPA. We also looked more specifically at different loci across the genome. We did that by CheapSeq. We had to do this in the whole worm, though, instead of looking just at the germline. And what we saw was very interesting. We saw a redistribution of K9 and K27 across the chromosomal landscapes. Especially, we saw a reduction of K9 and K27 at the ends of chromosomes. I'm showing these arrows point to 2 examples, but we see that across all 6 chromosomes in C. elegans, where there's a loss of K9 and K27 in BPA compared to DMSO at the ends of chromosomes versus the center of chromosomes, where in some cases we actually see a little bit more of the marks. Those effects actually more stronger, they're more significant at the ends in terms of chromosomal distribution, as opposed when you look at gene bodies where we did not see a difference with K9, and there's a significant reduction, though, of K27-trimethyl across gene bodies when the worms were ancestrally exposed to BPA compared again to DMSO and water. So what I've shown you so far are 2 independent correlations. I've shown you that a prior exposure, ancestral exposure, the P0 to BPA, causes a decrease in reproductive function. And I've also shown you that the same BPA exposure causes a decrease in 2 repressive marks. But these so far are 2 independent correlations. And we wanted to go beyond these correlations and show that this alteration of the epigenome was indeed required to see the reproductive defects caused by BPA. So to do this, we did an intervention. We exposed the worms at the P0 to DMSO control or BPA. And then only after the exposure was performed and we have, using the sensor, we knew that the worms responded to the exposure. Then in subsequent generations at the F1, we did RNAi, so a knockdown approach. We also used chemical approaches that I don't have time to show you here. And the goal by doing this intervention was to try to do genetically or chemically the opposite of what BPA did. So what we did was to target demethylases whose role is to remove demethylation. So if you follow the scheme here, what we did is knock down the demethylases that target K9-trimethyl and K27-trimethyl. And therefore, we promoted artificially through RNAi or, again, drug intervention, we promoted a state where we had more of these two marks retained on the chromatin. So I'm showing you here one way that we looked at this. This is by immunofluorescence and just some panels, but the full panels are available in the paper. I'm showing that by doing RNAi against GMGD2 here or GMGD3 UTX1 as a complex here, we can prevent the effects of BPA that reduces the mark. So we end up with more K9-trimethyl and more K27-trimethyl in those RNAi contexts. So now we can ask, what is happening to reproduction? Are we somehow reverting reproduction to normal levels? And the answer is yes. The effect is definitely more penetrant with GMGD3 UTX1 RNAi than with GMGD2, but either RNAi was able to remove the difference between BPA and DMSO in terms of embryonic lethality, as well as other features of reproduction that I don't have time to show here. So the question, of course, is why are we seeing germline apoptosis caused by this ancestral BPA exposure? And we know that, again, it's not just apoptosis. I've shown you effects on embryonic lethality. We also know that there's errors in chromosome segregation that is happening. So a large body of investigation right now in our lab is trying to dissect the mechanisms of this inheritance of reproductive dysfunction across generations. And actually beyond the inheritance, really understand what is going on that can trigger apoptosis. And we know that there's two ways of triggering apoptosis in the germline. One of them is altering chromosome dynamics, so the way that the homologs, the paternal and maternal chromosomes interact. And also the other way is to alter the process of meiotic recombination that leads to exchange of information across between the homologs. And these two processes are absolutely crucial for the germ cells to perform meiosis correctly, and they're highly, highly monitored by germ cells and by germline nuclei. And so we're investigating right now, and we have very strong evidence that one of these two processes, chromosome dynamics, is strongly altered, and that meiotic recombination may not be playing as much of a role in that process. So what I've shown you today is that BPA can cause transgenerational impacts on reproduction. Again, we also used a sensor, which I didn't have time to show you today. We've also shown that this is mediated by the female germline and not the male germline, and that this is also correlated with reduction in K9 and K27-trimethyl. Now, of course, we've gone beyond this correlation, and we've shown that this reduction in these two marks, K9 and K27-trimethyl, but especially K27-trimethyl, is very important for that transgenerational impact of BPA on reproduction and that germ cell loss. So I just want to end by saying that C. elegans is just one model that we use in the laboratory. We're also using other models that we think are highly complementary, and all of them are dedicated to understanding the mechanisms of inheritance of information across generations. So we're using C. elegans. Of course, we're using Daphnia magna, which is a small crustacean, which is used in transgenerational studies in ecotoxicological settings, but we're adapting this to our own settings as well, and doing comparative studies between the two. And of course, we have to be sensitive to the fact that C. elegans is not a mammalian species by definition, and so they are processes and they are epigenetic marks, languages, repertoires that are not accessible with C. elegans. And so We're also developing and adapting in vitro models of mouse germ cell development. So especially mouse pyramidal germ cells, that's a very extensive reprogramming of the epigenome, and we're monitoring how the environment may play into that reprogramming at all. So we're using BPA, but we're also using PFAS, alcohol, and inhalables as different chemicals of investigation. So I would like to thank, of course, my lab and the transgenerational team at the top here were fundamental in doing this work and following up on this work. My funding sources, I'm very active on Twitter. So if you'd like to follow our lab and any news from the lab, please follow us on Twitter. And especially important in this setting here with this recorded talk, if you have any questions or comments, you can scan this QR code here. It will send you to a form where you can enter, again, your comments or ask questions. And it's very important to me to respond to all these comments and questions, so we'll have the opportunity to interact there through this QR code and the form. So thank you very much. And again, I'm looking forward to any comments or questions from you.